We'll start today with the second part of the digestive system, which includes the small intestine and the large intestine. The small intestine consists of the duodenum, jejunum, ileum, whereas the large intestine we have the cecum, colon, rectum, and the opening to the exterior through the anus. Small intestine length, it's around and more than 6 meters in length, divided into 3 regions, duodenum, jejunum, and ileum, and the function ranges from hormone secretion to digestion and nutrient absorption. As a histology, it consists of 4 layers: tunica mucosa, tunica submucosa, tunica muscularis, and tunica serosa. The tunica mucosa, which is found near the lumen, which consists of the epithelium, lamina propria, muscularis, mucosa. The epithelium will be simple columnar epithelium with the goblet cells. The lamina propria will be the loose connective tissue, and the muscularis mucosa will be small layers of smooth muscle that separate the tunica mucosa from the tunica submucosa. The tunica submucosa consists of the dense irregular connective tissue, and the tunica muscularis will be 2 layers, inner circular and outer longitudinal. And finally, we have the tunica serosa, which will mostly be simple squamous mesothelial cells. Beneath these mesothelial cells will have small layer of the loose connective tissue, and these blood vessels mostly will be between the epithelium and the connective tissue. So this layer here, this is the layer of the tunica mucosa, submucosa, and then we have this muscle here, and the outer layer here will be the submucosa. As I said before, that the length of the small muscle, which include the duodenum, jejunum, and ileum, is around 6 meters. But this area is not enough for the process, as I said, hormonal secretion, digestion, and absorption. We have 3 modifications to increase the surface area. These modifications, we have 3 modifications. We have the plica circularis, villi, and we have the microvilli. These 3 together increase the laminal surface area, which is available for secretion, absorption, and digestion about 400 to 600 times. So instead we have 6 meters. If we multiply 6 by 400, that will be 2,400 or 3,600 meters. So imagine how much these modifications increase the surface area. So the first one, which is the plica circularis, or the other name, which is the valve of the Kerfring, these circulars or plica circulars are permanent spiral fold of the mucosa and submucosa. Permanent, they not disappear when the small intestine get filled with the food. They will be the same. This is different from the gastric fold that we saw in the stomach. These folds in the stomach not permanent, and they are disappear when the stomach is full. Usually these plica circularis is found in the duodenum, distal half of the jejunum, and the proximal half of the ileum. We can see this structure here. This is folds, circular folds from the mucosa and the submucosa. As I said, they are permanent, and this increases the surface area. See this one here, this here, till this area here, all of that, we call it as the plica circularis, which spiral permanent fold of the mucosa and the submucosa. Another modification, which is the intestinal villi or villi, which are finger leaf-like projection of the mucosa. They are projection only from the mucosa, and the appearance is like fingers, will be like this. These are called the villi. And mostly the core, the center of these villi will be the lamina propria. I will talk about it later. And it contains, because it's a loose connective tissue, they have the typical connective tissue cells, which is the fibroblast, mast cells, smooth muscle, capillaries, and also contain a blind end lymphatic channel, which we call it as the lacteal. See this one, this from here to here, this is the plexus circularis, like, but this one here and this one here, we call it as the villi or the villi. See, this is the finger-like projection. We call it as the villi. As I said, the center, this is the epithelium from outside. The center here, which is the lamina propria, which is typical as loose connective tissue, will contain fibroblast, mast cells, blood vessels, and also will contain a single blind end tube, which is the lacteal. Another modification we can see, which is the microvilli or villi. They are present in the area in the apical portion of the simple columnar epithelium. This structure here, the appearance like toothbrush, so that they call it striated border or the brush border. And all of these, only the name for the microvilli. And these are projection from the plasma membrane. Inside it, we have the actin filament to support these microvilli. See all of these structure here, all of these microvilli, and these microvilli are supported by the actin filament. As I said before, we have the tunica mucosa, submucosa, muscularis, and tunica serosa. So we start with the small intestine mucosa. The epithelial layer will contain different cell types. We have the enterocyte, which is main function for the absorption. We have the goblet cell, which is responsible for production of the mucus, and this is very important for protective and lubricant coating of the small intestine. As we go down from the duodenum to the large intestine, there is increase in the number of the goblet cells. Another cell we have is the enteroendocrine cells. As I said before, in the stomach we have the enteroendocrine cells, and these cells, it's very important because they produce hormones that stimulate or inhibit the digestion process. Very important cell only found in the small intestine, which is the Paneth cells. These cells contain granules, and these granules contain antibacterial agents and also play important role in the small intestine protection. So we've started at the epithelium. As I said, we have a tall columnar epithelium. Between these columnar cells, we have the goblet cells. As you can see here, we have number of the simple columnar, which is the enterocyte, and between them we have scattered goblet cells. As I said, we have the goblet cells inside here. We have the granules, and these granules contain the mucinogen. Once released, the name will change into mucin, which is the active form, and this will be responsible for the coating and lubricating the surface of the small intestine. As you can see here, this is the simple columnar cells, and between these columnar cells, we have the goblet cells. Again, the simple columnar goblet cells. And this layer just below the epithelium, which is the layer of the lamina propria, which contain the different cells of the connective tissue, also the blind in the tube of the lymphatic vessels, which is the lacteal. In the base of the epithelium near the muscular mucosa, we have, and we'll see here, there are some cells that contain apical granules. These cells are the Paneth cells. As I said, they reside in the bases of the intestinal crypts. They are secretory cells. They are responsible for protection of the antibacterial, antiparasitic agents. So they help against bacteria, also the parasites. And you can see here, this is a scanning electron microscopy. This is the location of these glands. They reside in the base of the intestinal glands. Again, the cells, as we can see, almost pyramidal in shape, and we have these granules. Again, this is the Paneth cells. As I said, these cells only found in the small intestine. We could not find them in the stomach, or we could find them in the large intestine. Another important cell is found, the regenerative cells, which is the intermediate cells. These cells found in the lower half of the crypt, or intestinal crypt, and these very important because these cells can differentiate into enterocyte or the goblet cells. Now we have very important term, which is the intestinal gland, or another name, which is the crypt of the Lieberkuhn. This extends here from the middle of the villi toward the muscularis mucosa. From this area here to this area here, we call it the intestinal gland or the crypt of the Lieberkuhn. These cells contain the enterocyte, the goblet cells, we have the regenerative cells, and very important, near the base, we have the Paneth cells, which is very important as antibacterial, antiparasitic cells through the lysozymes they secrete. Again, from this area here to this area here, we call the intestinal gland. This is almost from the middle of the villi or the villi toward the muscularis mucosa. For the lamina propria, which is occupy the villi course of the intestine between the crypts of the Lieberkuhn, usually they are loose connective tissue. Again, they contain the typical lymphatic connective tissue cells, which is the mast cell, fibroblast, smooth muscle, nerve ending, and very important, they contain the lacteal, which is the lymphatic vessels. So this is the epithelium, and this the layer just below it, this is the area of the lamina propria, which is the loose connective tissue. There is important glands that found in the duodenum. These glands are called the Brunner's gland, only found in the duodenum. So this is a landmark for the duodenum. These glands typically branch tubulo-alveolar glands produce alkaline secretion. Remember the food that's coming out from the stomach will be acidic chyme, will be acid. So this acid can burn the mucosa of the small intestine. As protection, we have these Brunner glands secrete. This is the gland found in the submucosa, secrete their product and protect the luminal area of the duodenum, because the secretion of this gland contains a lot of alkaline secretion, and this is very important to neutralize and protect the small intestine from the acidic chyme. These alkaline secretion, neutral and alkaline glycoprotein, and contain biocarbonate. All of these very important to neutralize the acidic chyme. As I said, these glands are found in the area of the submucosa. This is the mucosa. This is the muscular mucosa. This is the area of the submucosa. And the gland only found at this area here. All of these glands are called the Brunner's gland, and this gland only found in the duodenum. So we'll talk about a little bit about the landmarks of the small intestine, duodenum, jejunum, and ileum, how we could differentiate with them. Very easy. For the duodenum, we have short villi, but importantly, we have the submucosal gland, which is the Brunner's gland, as I said, only found in the duodenum in the area of the submucosa. Jejunum, they have very long villi, There is no Brunner's gland, and as we can see here, because these villi or villi very long, when we cut it in the microtome, they appear as separate parts, and these separate parts they call it the float island. Okay, float island because they appear as island that surrounded by the sea, and you can see here there's no any connection. And very important, there's no gland in the submucosal. The last organ, which is the ileum, again we have the villi. We have very elongated villi, float island, but very important, in the area of the submucosa we have aggregation of the lymphatic nodules. These aggregated lymphatic nodules, they call it as Peyer patches, and they only found in the submucosa and the submucosa of the ileum. So the duodenum we have in the submucosa Brunner's gland. In the ileum we have the Peyer patches. In the jejunum we don't have either the Brunner's gland or the Peyer patches. I'll go back a little bit for this slide here. So this is the area of the submucosa, which is a dense irregular. This is the area here where we have the muscle. The muscle are two layers. We have the inner circular and we have the outer longitudinal muscles. Okay, see, inner circular and outer longitudinal. And the area here, which is the area of the serosa, usually and typically we have simple squamous epithelium, and beneath the squamous cells, we have a layer of the loose connective tissue. Again, here we have the muscles. We have two layers. We have the inner circular and the outer longitudinal, and typically at this area here we have the serosa. When we consider the cecum and colon, their primary function in the water absorption and electrolyte, very important, there is no villi in the cecum or colon. Their epithelium, typical epithelium, are the simple columnar epithelium with the goblet cells. Again, we have the absorptive cells, regenerative cells, and we have some enteroendocrine cells. We have also the layer of the lamina propria, which is just beneath the epithelium, which is loose connective tissue. There is no Paneth cells. In the submucosa, we have fibroelastic connective gland, connective tissue, and there is no gland in the area of the submucosa. We can see here, this is the typical organ. We have the area of the mucosa. We have muscularis mucosa, and then we have the area of the submucosa, and then we have the area muscularis externa, and finally we have the serosa. Typical cells, we have the absorptive cells, which is the simple columnar epithelial cells. We have the goblet cell, we have the regenerative cells, and we have some enteroendocrine cells. And this gland from here to here, we call it as the intestinal gland. For the muscularis externa, again, we have two layers. We have the inner circular and the outer longitudinal layer. But very important in the cecum and colon, the outer layer, which is the outer longitudinal, the muscles gather into three longitudinal smooth muscle band, and we call it the tinea coli. And if you remember for the anatomy, we talk about the large intestine and the cecum, and we talk about this band and how we can differentiate between the different parts of the colon and the cecum. Again here, this is the area of the mucosa. We have the epithelium, and we can notice the amount of the goblet cell, the increase in the number of the goblet cells. Between these, we have the lamina propria, and then we have the muscularis mucosa, and this is the area where we have the submucosa, And then we have the inner circular. And notice here, this area here, the collection of the outer, instead of spreading around the wall of the large intestine, only we have gathered into bands. These bands, which is called the tinea. Again, this is the epithelium, and all of these cells are the goblet cells. And between them, we have the lamina propria, we have the muscularis mucosa. From this area here, we have the area of the mucosa, and just below it, we have the submucosa. We can see here that muscularis mucosa as it separates between the mucosa and the submucosa. The last portion, cecum, colon, then we have the rectum, similar to the colon, however, contain fewer and deeper crypt of the Lieberkuhn, which is the intestinal glands. And then we reach to the level of the anal canal. It will consist of the anal columns, and between these columns, see this area of the column, between these areas we have the anal sinuses. Usually the epithelium changes from simple columnar epithelium into stratified cuboidal, then stratified columnar, then it will start to change as we reach near the area of the anus to non-keratinized stratified squamous epithelium, and the final area, which is the area around the anus, will be keratinized stratified squamous epithelium. So these areas here are called the anal column. Between these anal columns, this is the anal sinuses. The epithelium starts with simple columnar, then start to change stratified cuboidal columnar, then change into non-keratinized stratified, and when we reach to the level here, which is the area of the anus, start to be keratinized stratified squamous epithelium. As you can see here, this is the rectal junction. This is the area of the anus, and this is the area of the rectum. You can see here the anal columns. Between the columns, we have the sinuses. Epithelium change from simple columnar into stratified cuboidal, columnar, then non-keratinized stratified, then keratinized stratified squamous epithelium. Now we'll talk about the neural elements of the small intestine and the large intestine. As we know, the gut is more or less autonomous. The control and the function, because all of the wall of the small intestine are smooth muscle, and the control of these is through the autonomic nervous system. However, the central nervous system can override that. The enteric division of the nervous system, where CNS, central nervous, send extrinsic preganglionic fibers, most control in the local but can be overridden, control feedback loops with sensory and motor function, because as we know, our brain should know exactly what's going on in the small intestine or large intestine. Mostly consist of two neural elements. One of them found in the submucosal, we call it the submucosal plexus, and this controls the smooth muscle of the area of the muscularis mucosa. And the second one, which is the myenteric plexus, and this will control the smooth muscles that found at the area of the muscularis externum. So then the first one, the submucosa, as I said, it's found in the submucosal tunica. As I said, it controls the contraction of the smooth muscle in the lamina propria, which is the muscularis mucosa, this area here, and this here where we have the fibers. And usually this is very important because we need the food for mixing and movement through the GI tract. As I said, these are the cells with their fiber. The second one, which is the myenteric plexus, as I said, it's found in the area between the two layers, the inner circular and the outer longitudinal. This here, this area here. And this is very important because they control the contraction of the smooth muscle here, and they are responsible for the peristaltic movement of the small intestine and large intestine. The myenteric plexus, as I said, this is scanning electron microscopy, but here in the lab, if we are going to find these cells, which is the myenteric plexus, very easy because we have to go between the two layers, the inner circular and the outer longitudinal.